Okay, question 11. We have carbon dioxide and growth. Um, the correct answer here is C. When you look at this graph, we see that adding extra carbon dioxide increases the total weight of the dried plants grown in ideal versus stress conditions. And then it's asking, based on the figure, which statement describes the observed relationship between CO2 enrichment, which is the gray versus the black, and plant growth under ideal and stressed conditions. So it wants you to explain the whole thing. Now, the correct answer is that if you increase the atmospheric CO2, so that's the black line, it results in a greater increase in plant growth under stressed conditions, which it does, an increase of about 0.4, whereas the increase here is an increase of about 0.2. So many of you got it wrong because you selected B. Actually, everybody who got it wrong selected B. The increase in CO2 results in a greater increase in plant growth under ideal conditions than stressed conditions. And I want you to pay attention, read that wording carefully. The increase in the atmospheric CO2, so that's comparing gray to black, not ideal to stressed, right? This would be correct if it said ideal conditions result in greater plant growth compared to stressed conditions. That is true. But they're specifically talking about the change in the amount of carbon dioxide. So changing the amount of carbon dioxide did not make the ideal plants grow less successfully. It didn't make the stressed plants grow less successfully. They both were more successful when carbon dioxide was increased, but stressed plants were even more successful. Not more than ideal, but more. They increased it they increased more relative to a lower carbon dioxide level in the atmosphere. Now this question looks at phosphorus levels and how it affects growth. This is pretty much straight. Can you identify the correct answer as a result of which elements are found in each of the major macromolecules? You got to either be able to look this up or you need to have it memorized. Phosphorus is a critical ingredient in the backbone of DNA. It also forms the phosphate heads on many of the different kinds of lipids. Some proteins have phosphorus in it, but it's much less critical to the formation of proteins. Of the answers here, B and D cannot be correct because they both reference carbohydrates, which have no phosphorus in them, so the plant doesn't need them to make carbohydrates. A and C are a little tricky. C is the better answer because nucleic acids need phosphorus. It's a critical requirement. Most proteins don't contain phosphorus containing amino acids. If you selected A, there was one of you. This is not a bad answer. I kind of don't like this question. I feel like it's a little misleading. C is better because it is nucleic acids and lipids that would be more impacted. It was only one of you that picked A. I don't know if it was because you were thinking of proteins or because you just misunderstood the question overall. But for the majority of you who got this one wrong, you need to be able to connect elements to the macromolecules. Question 13. So we have dehydration synthesis. Let's see. As an amino acid must have which of the following properties in order to be incorporated into a polypeptide? And a polypeptide means we have a new peptide bond forming here between the OH group of the carboxyl group and the amine group of another molecule. So the ability to form a covalent bond with its NH2 group and its COOH group. So a new amino acid would have its NH group and the COOH group here. The central carbon doesn't react with the nitrogen to form the peptide bond. The central carbons are here, and here. So this is, again, structure function. You should be able to draw out what a polypeptide looks like. You should be able to kind of maybe even diagram how a new polypeptide would come in. I would be able to do that for each of the major macromolecules. So for nucleic acids, I would be able to draw out a monomer or a polymer, and I would show how a monomer would attach on the, which end is going to attach on the 5', the 3'. I would be able to do it for amino acids and show where the R group is in the central carbon. I'd be able to do it for carbohydrates and show the ring structure of glucose molecules coming together. What is the polysaccharide versus the monosaccharide? Okay. Just know your way around those diagrams because you're going to see diagrams. Which of the following is a common feature of the illustrated reaction showing the linking of monomers to form macromolecules? Most of you selected the correct answer here. Could state that monomers use covalent bonding to form polymers. Which is interesting because that's basically what this question was. It was just the details of exactly where that bonding happens. Okay, so looks like a lot of you understand the big idea, maybe need some help with the more specific idea. Which of the following describes a key difference among the 20 amino acids? C is true that some amino acids are hydrophobic. Only some have a carboxyl group. That's not true. They all do. Only some have an R group. They all have an R group, right? Some amino acids contain the element phosphorus. That's not a key difference. The fact that they are hydrophobic is a key difference, or hydrophilic, or so on and so on. Amino acid substitution shows amino acids are part of a polypeptide chain, showing the same section of the chain after a mutation has occurred. So we have aspartic acid, cytosine, and lysine versus aspartic acid, valine, and lysine. And the question is here, how might this change the structure and function of the protein? Again, this is primary, secondary, tertiary. The R groups of the new amino acids have different chemical properties than the R groups of the cytosine. This will change the protein to misfold and not function properly in the cell. That's true. The new amino acid valine has replaced cytosine in the new protein. Well, that's true. Since the number of amino acids have remained the same, there will be no change in the three-dimensional folding. That is not true. So you need to understand that these R groups are going to change the way it folds, right? Since the new amino acid is bonded on one side by an amino acid which is negatively charged R group and an amino acid on the other side which has a positively charged R group, and that's true. There's a negatively charged R group and a positive, which means they're going to be hydrophilic. The charges will balance and the protein will fold as usual. That is not true. If this one was also hydrophilic, that would change the shape of this. They don't balance and then cancel this one out. This one still has its own R group that's going to interact in its own way. Okay. 17. What we got here? Okay, we got two more. So we have a segment of a DNA molecule. It looks like a hot mess, but that's because they're showing the bases in here with their hydrogen bonding. Here we have the sugar phosphate backbone. Which of the following statements is correct about the molecule shown in 1? It's DNA because of the nucleotides present. Well, we have CGAT, okay? And some of you said it's DNA because of the nature of the hydrogen bonds between the guanine and the cytosine. Nope. GC bonds happen in DNA and RNA, and there's always 3 of those hydrogen bonds. That's the bonds here. AT bonds have 2. AU bonds also have 2. So the hydrogen bonding, which is what holds the DNA together, is always intact. Which of the following best describes the structural similarity between the two molecules shown that is relevant to their function? So both contain nucleotides that form bases with other nucleotides, which allows each molecule to act as a template in the synthesis of other nucleic acid molecules. True. That's how DNA is used to make mRNA, or mRNA is used to connect to tRNA, and so on. Some of you said both contain nitrogenous bases and phosphate groups. which allows each molecule to be used as a monomer in the synthesis of proteins and lipids. So they do both contain nitrogenous bases and phosphate groups. That's true. But they're not used to build proteins and lipids directly. That's the process of the central dogma, so transcription and translation, that it's referencing there. Okay, so those are the questions on this. Let me know if I can help with something.